Our visualized protocol provides a frame of reference for wider research on defenses in fungus-growing ants. The main advantage of this approach is that it will help researchers recognize key defensive behaviors to ensure a common understanding for future work. A reference for research in fungus-growing ants is likely to be particularly useful for young researchers who are not familiar with these behaviors. A number of these behaviors are infrequent, so experienced researchers may never have observed them. This guide will facilitate comparisons across studies and ant species by illustrating key defensive behaviors along with consistent definitions. Furthermore, understanding behaviors in controlled conditions may help studies in natural habitats where conditions are hard to control.
To begin, isolate Escovopsis strains from Acromyrmex echinaceae laboratory colonies by placing fungus garden fragments with ants removed on a petri dish with moist cotton wool for several days until Escovopsis germinates and sporulates. Transfer spores to potato dextrose agar plates and incubate at approximately 23 degrees Celsius for 2 weeks. Using a sterile needle, remove mature spores from the plates. Then, inoculate the spores on new plates under sterile conditions and incubate them at approximately 23 degrees Celsius for 2 weeks. When the hyphae have grown to cover the entire plate and into mature brown spores, inoculate the spores onto new plates under sterile conditions.